नमस्कार दोस्तों, welcome to channel Toxicology on board. In this video, we'll be discussing another DABT topic, that is mouse lymphoma assay, forward mutations in mammalian cells. Mm, uh, Again, you have to remember, for the DABT syllabus or examiner, no, genotoxicity mutations, these are the very important topic. In my channel also you might have come across the various types of battery of genotoxicity tests, and which type of assay is detecting what type of DNA damage, whether it is point mutations or forward mutations or chromosomal damage. So there will be different type of assay and specific type damage, DNA damage it is identifying. So that you have to remember. So from that point of view only, examiner will frame the questions. So in this video, we will discuss about mouse lymphoma assay, forward mutations in mammalian cells. So let's directly dive into the question. The question is, what genotoxicity endpoint can be detected by mouse lymphoma assay? Direct question. If you know the principle, what is this assay it is giving, then you can easily answer this question. So let's see what are the options we have. We have option number A, that is germ cell cytogenic changes. Option number B, unscheduled DNA synthesis. Option number C, which is micronuclear degradation. And option number D, which is forward mutations in mammalian cells. The right answer will be in the green text, that is option number D, that is forward mutation in mammalian cells. So please look into the explanation and the reference which is given by the American Board of Toxicology. The mouse lymphoma assay identifies evidence of, so we have four things. It is identifying point mutations, deletions, translocations, and recombinations. Okay, these are the four type of things that can be identified by this mouse lymphoma assay. What are those? Point mutations, deletions, translocations, and recombinations. So while reading this question, also read ICH guideline. We have S2 sections में A, B, C are there, which are specifically for a genotoxicity and mutation type of damage, genetic damage के लिए. So in that you read about S2B. S2B में what it is, the name of the guideline, a standard battery of genotoxic test for pharmaceuticals. Okay, so in that also ABT is asking us to go and read in detail section number 6. So next slide I will be having what is section 6 I will be discussing, that to note to. And also in addition to this guideline, they are also referring us to go back and read Cassett and Dow 9th edition, page number 468. So you can quickly pause the video and also read in this guideline also the basic principle, what is the, how this mouse lymphoma assay is performed, that also you read. Okay, so that again any question with respect to principle of this assay you'll be able to answer in the exam. So let's directly see what is this section 6 in the ICH 2B guideline refers to. The molecular dissection of the mutants induced at the TK locus. So in the DNA also different locuses will be there. No, particularly at the TK locus shows a broad range of genetic events takes place. What are those events? Including point mutations, deletions, translocations, and recombinations. Okay. Small colony mutants have been shown to predominantly lack the TKB allele. So whenever there is a small colony mutants are there, no, particularly this particular TKB allele, it is lacking as a consequence of structural or numerical alterations or the recombinational events. Whenever there is a chemical is there, chemical is getting exposed to this particular DNA at this particular allele TKB, there is a lack of expression is taking place, there is a aberration is taking place, because of which okay, so there is some evidence that other loci apart from this TKB allele such as HPRT or GPT are also sensitive to large deletion events. However, due to X chromosomal origin of the HPRT gene, which is probably flanked by essential genes, large-scale deletion events or numerical alterations often do not give rise to mutant colonies, thus limiting the sensitivity of the genetic locus relatively to the this particular allele, TKB allele, TK locus, or for the detection of wide range of genetic changes. So this is the mechanism or the background behind this particularly this one. Okay, TKB allele. Okay. See, let's understand in detail more. The mouse lymphoma assay is a mutagenicity assay used to determine whether a chemical is capable of inducing mutations in eukaryotic cells, not prokaryotic. Okay, typically mouse lymphoma L5178Y cells are used, and the ability of the cell cultures to acquire resistance to thymidine, the result of forward mutation of the at the thymidine kinase TK locus, this one is quantified. So here the damage it has been quantified, which becomes the primary reason for this particular mouse lymphoma L5178Y mouse lymphoma SA. Okay. So, what are the additional things in that section 6? We will see the mouse lymphoma assay is a genotoxic test used to detect mutations in the thymidine kinase TK gene of mammalian cells, not prokaryotic, eukaryotic. It is widely used for regulatory purposes, so it is a requirement apart from AIMS assay, chromosome aberration assay. Particularly this mouse lymphoma assay is also a regulatory requirement either from the FDA or the EMA to assess chemical potential to cause genetic damage, and it can detect both point mutations, that is large colony mutants, and chromosomal damage, small colony mutants. So by this assay both the things can be identified, that can be detected. So this mouse lymphoma assay can be performed in two versions using agar based and microwell based method. Okay, there are two methods are there for this one that you will learn in the case of principal. What the different steps are there? Let's see. Cell line. The test uses a specific mouse lymphoma cell line that is heterozygous for the TK gene, TK plus as well as minus trait, meaning it has one functional copy. Mutagenesis. The cells are exposed to the chemical being tested. Okay, the chemical which you are testing. If the chemical is mutagenic, it can cause a mutation in this functional gene. TK gene is there, no, making the cell unable to produce the TK enzyme. Now the chemical is there, the cells are there in which TK gene is active. Whenever this chemical is exposed to these particular cells, this TK gene becomes inactive and it is unable to produce this TK enzyme, thymidine kinase enzyme. Selection. The cells are then grown in a special medium containing TK inhibitor. Then whatever the cells are there, no, uh, such as what is this TK inhibitor? 5-bromodeoxyuridine, that is BRDU. Only the cells that have lost TK enzyme, the mutants can survive and grow. Okay. So if you are using a TK inhibitor, the cells will unable to grow, but particular this enzyme in which the enzyme is already been lack of enzyme is there in that cells, they are only able to survive. Only the cells that have lost the TK enzyme, the mutants can survive and grow. This is the basic principle. Okay. So, what are the classification of the mutants? The surviving mutant colonies are classified into two types: large colony mutants, small colony mutants. Because of which we have learnt in the previous slide that it can identify both chromosomal aberration type of damage, means the small colony mutants as well as at the chromosomal level as well as large colony mutants. These typically result from point mutations and indicate a gene mutations, whereas the small colony mutants, these are associated with gross chromosomal damage such as deletions or translocations and indicate a chromosomal mutation. So what are the data analysis? Researchers then analyze the frequency of both large and small colonies to determine the mutagenic potential and mode of action of the test chemical. Okay, so what are the applications of this mouse lymphoma assay? So as we have seen that it is used for regulatory testing, it is used for predictive testing, mechanistic insights, alternative animal testing. Let's see what is the regulatory testing. The mouse lymphoma assay, it is a standard test in the core battery of genotoxic test. In addition to micronucleus assay, AIMS assay, chromosome aberration assay, this mouse lymphoma assay is important, required for the registration of the pharmaceuticals, pesticides as well as chemicals. Predicting toxicity by providing information on both gene and chromosomal mutation. This assay helps in the assessment of substance potential to cause cancer or other diseases. Okay, so once there is a damage in the case of DNA, there is a potential for that chemical to cause cancer. Mechanistic insights. The distinction between the large and small colony mutant provides insight into the mechanism of this chemical genotoxicity. Unlike other assays, no, this is able to detect at both chromosomal aberration level as well as point mutations level, no, because of which it is able to identify both large colony mutants as well as small colony mutants. So this helps us better understand the mechanism how the chemical is causing the particular genotoxicity. Coming to alternative to animal testing, this assay is an in vitro test. Okay, meaning it is performed in a lab setting and helps the risk assessment process before any in vivo animal testing is conducted. Before going to micronucleus assay in the mice or rodents like that, you can always go for mouse lymphoma assays because this is an in vitro assay. So these are the important take home points. with respect to mouse lymphoma assay. So very important. You have to read about the genotoxicity, battery of genotoxicity assay, and list of genotoxicity assays are already given in the class and textbook. Okay, read ICH S2B S2 series of guide ABC are there, no, which is with respect to genotoxic mutagenicity from the exam point of view. So thank you so much. Be curious. For any questions, please comment arya.priyanka18 at the rate gmail.com. See, the only tip for the preparation or understanding the genotoxicity is that you have to keep read. See, those people are already working in genotoxicity as a study director, no, or those who are working risk assessment of genotoxicity. For them, it is very easy. They can know which is the assay, which is causing which type of damage, what is the principle, what is the method. Those who are working in other domains, somebody who's working in acute six-pack studies, or somebody who's working in cosmetic or agrochemical, they might not be aware of this type of genotoxic testing. So, what you have to do is just the only tip is that you keep reading, make flash notes, essay, the inference, what we are getting, principle. These are two, three things are there. You make a notes, keep reading. Repetitive reading is the only way to memorize this thing. Because in the exam also you have to understand, you have when you have when you are in the aptitude exam, no, timing is important. So straightforward questions. If you look into the first slide, no, this is a direct question. Mice lymphoma detects what type of mutations. That's all four are there. Small colony, large colony mutants it is doing. Point mutations, chromosome, all these things it is doing. But you should be knowing which essay. So straightforward questions, can you should not be wasting any time. Okay, so that can be only done by repetitive reading. If you're already working in that area, it's well and good. You will be able to remember. But though, because since toxicology is a wide discipline, there are different areas, people are there differently they are working. Somebody might not be working in the genetic toxicology area. For them, it is very difficult to remember. So the answer is only to repetitively keep reading those tables which are given in the lesson or make flash notes, and that's the thing. Okay. So thank you so much. Subscribe, like, subscribe, and follow my YouTube channel, Toxicology on Board. Thank you so much. All the best for your exam.